The central dogma says genetic information must move from DNA to proteins through transcription and translation. There are 3 steps to transcription. In the first step, an enzyme, RNA polymerase, synthesizes a sequence of RNA nucleotides along DNA template strand. The second step makes the RNA strand have complementary nitrogen bases to the DNA template strand. Uracil is used instead of thymine in RNA. Finally, the RNA strand leaves the DNA template strand. The DNA repairs and winds up.
This new RNA strand goes through RNA splicing now. Here, all the introns are cut out of the RNA, leaving only the exons. This is the mRNA. tRNA is a transfer of RNA. It has the start codon AUG and a stop codon UAG on the end. There's 3 bases on tRNA called the anticodon. The anticodon pairs with an mRNA codon with the ribosome binding site.
Translation moves the genetic information on the RNA to the protein. The mRNA strand travels on the ribosome. There are 2 ribosomes, the large and small subunits. On the ribosome are an A site and a P site. The A site holds the tRNA, which is carrying the next amino acid to be added to the chain. The P site holds the tRNA that carries the growing polypeptide chain. The tRNA with an amino acid attaches to the small subunit, forming an initiation complex. The initiation complex binds to mRNA and travels down until it reaches the start codon. The anticodon and tRNA form base pairs with the start codon.
The large subunit attaches to the small subunit with the mRNA in between. The start tRNA is in the P site on the large subunit. Another tRNA enters the A site on the large subunit. The amino acid of the first tRNA moves to the catalytic site, which helps it bind to the amino acid of the second tRNA. Peptide bonds begin to form protein.
The leftover tRNA is let go and the ribosomes move down the mRNA one to the right. The tRNA that's attached to the 2 amino acids is now in the first binding site and the second site is empty. A new tRNA enters the second binding site with its attached amino acid. The amino acid chain moves to the catalytic site where it's attached to the tRNA in the second binding site.
A new peptide bond is formed. The empty tRNA in the first site leaves and the ribosome moves 1 codon to the right. This continues until you reach the stop codon. The protein is released. The mRNA is released and the smaller and large subunits separate.